This video will look at metabolic rate in different animal groups and their different types of circulatory systems, and it's also going to look at the equipment used to measure metabolic rate. So metabolism is the rate at which the body uses energy, and our body releases energy by breaking down food or breaking down glucose and using oxygen for respiration. And when we do aerobic respiration, we produce carbon dioxide, we produce water, and we also produce energy, and we'll use that for things like movement and repairing cells. And if you're an animal that your metabolic rate is really high, it means you use energy really quickly, and if it's low, you use energy more slowly. So to compare metabolic rates in different organisms, we can measure things like how much oxygen they use, how much carbon dioxide they produce, or how much heat they give off. And we're going to use tools such as respirometers to measure oxygen consumption or carbon dioxide production. We can also use oxygen or carbon dioxide probes. And for heat production, we can use calorimeters. And these measurements are going to help us understand how fast metabolism is happening in an organism's cells. So this is a simple calorimeter, which is used to measure heat produced by an organism over a period of time. And we need to ensure that we are saying that this is over a period of time, as this is going to ensure validity so we can compare between different organisms. In this calorimeter, an organism is going to be placed into this little chamber here. And as it respires, it's going to give off heat, and that heat is going to be transferred into the surrounding water. And the temperature change is going to be detected by the thermometer here, and we measure that and use it to calculate energy expenditure or metabolic rate, and we can compare that with another organism or in a different environment. So the other piece of equipment that we need to be aware of is a respirometer, and a respirometer is measuring oxygen uptake or CO2 output of an organism over a period of time. So what we have here is a woodlouse that's been placed into a sealed container with air inside, and the sealed container is connected to this capillary tube, which is connected to this coloured liquid here. Now, as this woodlouse respires aerobically, it is going to be consuming oxygen and giving out carbon dioxide. Now, as it consumes that oxygen, it is going to draw this coloured liquid up the capillary tube, and the faster or the greater distance this coloured liquid moves, the greater the rate of oxygen consumption, so the higher the metabolic rate. Now, it's also going to be producing carbon dioxide gas, so we need something to remove that from the air. So we have these soda lime pellets, so we can tell that it is just oxygen uptake being measured here. And we'd be able to compare this across different situations. We could maybe have another respirometer set up at different temperatures, for example, and we'd be able to compare metabolic rate. So here we can see another respirometer that is being used to look at the effect of temperature on the metabolic rate of a snail. And with all these experiments, we should be able to identify the independent and dependent variable. And remember, your independent variable is the thing that you are changing in an experiment, and the dependent variable is the factor that you are measuring. So here our independent variable is going to be temperature, because that is the thing that we are changing. We're setting up this respirometer at different temperatures. And our metabolic rate is going to be our dependent variable. That is the thing that we are measuring and comparing. And what we should see in this experiment is that at low temperatures, its oxygen consumption is going to be slow, or it's going to have a low metabolic rate. And as the temperature rises, that oxygen consumption is going to increase. So we're going to see that coloured liquid moving further up this capillary tube here. And this is because metabolic processes are controlled by enzymes, so they should increase up until a point where the enzymes become denatured. So now we're going to look at different circulatory systems that we find in different groups of animals. And the first one we should be familiar with because it's the type of circulatory system that we have. So we find this double complete circulatory system in birds and mammals who have really high metabolic rates. They need loads of energy to survive and stay active for things like escaping predators. And they're also regulators, which means they have to maintain their internal environment regardless of what their external temperature is. And producing this constant heat requires loads of energy. We're going to look at that in another topic. So a double complete circulatory system is when the blood is going through the heart twice for every trip around the body. That's what we mean by double. In our first loop, we've got deoxygenated blood being pumped out of the heart, and it is going to the capillaries in the lungs where it's picking up oxygen. It's then returning oxygenated blood back towards the heart, where that can be pumped at really high pressure and delivered to the cells within our body. Then our deoxygenated blood is being delivered back into the heart, again where that can be pumped to the lungs where it can pick up more oxygen. So complete just means that the oxygenated and deoxygenated blood are kept completely separate. They don't mix because the heart has two sides, or two atria and two ventricles. So a common question here is, why is this type of circulatory system the most efficient? Or why does it support birds or mammals that have this really high metabolic rate? And we say it's extremely efficient because there's no mixing of oxygenated and deoxygenated blood. We're also pumping oxygenated blood out of the heart at a really high pressure. And it's really efficient because the body's always getting blood that's full of oxygen. We've not got any mixing there. So that's going to help keep the energy levels high. And that makes sense because mammals and birds often have really long distances to migrate, so they need to have these efficient circulatory systems. So the next type of circulatory system is a double incomplete, and we find this in reptiles and amphibians. And in this type we still have two atria, but we only have one ventricle. So double still means that the blood is passing through the heart twice per circuit. We can see that here. So we've got our blood being passed to the lungs or the skin in amphibians. We're picking up that oxygenated blood and we're taking it back to the heart. But because we only have this one ventricle, we're getting a mixing of oxygenated and deoxygenated. That's what we mean by incomplete. So that mixed deoxygenated and oxygenated blood is being pumped round the body to the cells, and it's going to be less efficient because there's going to be less oxygen in that blood. And again, that deoxygenated blood is going to be passed back into the heart, where again it's going to mix in the ventricle. So as a result, it's going to be less efficient than the complete system, but it works for animals with lower metabolic rates. So things like your reptiles and amphibians, they don't need as much constant oxygen and energy as birds or mammals. And I always think about Steve Irwin wrestling with crocodiles. You'll have seen that the crocodiles, they can only sustain a really high amount of energy for a short period of time. So they'll charge and then they'll kind of lose their energy. And that is because of this less efficient system. So last one that we're going to look at is the single circulatory system, which occurs in fish. And single just means that the blood is going through the heart once during a full circuit of the body, and incomplete means that the heart only has two chambers. So we've got an atrium and we've got a ventricle. So pathway of blood is starting at the heart, and the heart is going to pump this blood at really high pressure towards the gill capillaries, and this is where it's going to pick up oxygen that can be delivered to the body capillaries. So we need to think about what's happening in terms of pressure here. The heart is pumping the blood at high pressure towards the gill capillaries, and as the blood is squeezed through the tiny blood vessels, the pressure is going to drop. Now what that means is the blood is going to be moving much more slowly towards the body capillaries and at this much lower pressure. And as a result, we're going to be delivering oxygen a lot more slowly and a lot less efficiently than what we would in the complete circulatory systems. And this is okay for things like fish because they're cold-blooded. They don't need loads of energy to stay warm. So again, we're going to have low pressure in the body capillaries, and we're going to be delivering that deoxygenated blood back towards the heart. So in summary, the pressure is going to be higher in the gills, and it is going to be lower in the body capillaries or the body tissues. So just a quick summary here. In birds and mammals, we had a double circulatory system, so that meant blood was passing through the heart twice per circuit. And we said it was a complete circulatory system because we had two atria and two ventricles. There was no mixing of oxygenated and deoxygenated blood. As a result, it was the most efficient and supported animals with high metabolic rates. Amphibians and reptiles still had a double circulatory system, so blood passing through the heart twice per circuit, but it was incomplete because we only had one ventricle, so oxygenated and deoxygenated blood were mixing. As a result, this was less efficient and more suited to animals with lower metabolic rates. In fish, they only had a single circulatory system, so consisting of one atrium and one ventricle. Blood was only passing through the heart once per circuit, and we needed to be aware of the pressure changes. So he said it was going to be high pressure in your major blood vessels. So blood being pumped away from the heart towards the gills was going to be travelling at high pressure. As the blood passed through the gills and was squeezed through those tiny capillaries, it was going to lose pressure. So when we got to the capillary beds in the body, the blood pressure was going to be much lower, so much less efficient.